good afternoon, good morning, good evening. Welcome to online lecture 4D here in your Biology 140 semester. Here in online lecture 4D, we will be getting into the latter parts of Chapter 6. So we're looking at slides, maybe 18, possibly as far as 31. Probably, I don't know, we'll kind of see how it goes, guys. I don't want to overwhelm you here. We may have a go ahead and do a 4E, but let's go ahead and see what we can do here in 4D and continue to looking at this subject of population ecology. So we'll be discussing population ecology. We'll be talking about what affects population's growth, what causes populations to grow or to decline, and then we'll look at survivorship curves, look at different strategies that different species employ. So let's go on without further delay with all the slides we've got to cover. Let's go ahead and begin here on slide 18. So we're looking at factors that affect growth rates. So let's go ahead and look at what kinds of things affect the growth of a population. So we have births, immigration, deaths, and emigration, where R is the rate of growth. And so the rate of the population's growth would be births plus immigration minus deaths and emigration. So B and I cause the population to increase, right guys? And then D and E, deaths and emigration, cause the population to decline. And when you factor all these four items together, you get your overall growth rate, little r. So again, births, the number of births that occur in the population at any given time. And of course, this is going to vary greatly depending on species and with stress and food availability. For example, if it's a mouse population, well, mice are known to have lots of babies at a time, say 6 baby mice in a litter easily. And if there's plenty of food and not too much stress, not too much overcrowding, wow, it's extremely high birth rate. Contrast that with elephants, who have an 18-month gestation period, sometimes longer, and who typically only give birth every 5 to 6 years. So for the elephant population, it's going to go a lot slower, even if conditions are ideal. And of course, if conditions are very stressful, even for those that have lots of babies in birth and have high birth rates, you'll even see that drop if they are stressed. So even a mouse population can drop down in their birth rates just because of the lack of food availability, etc. Immigration, the number of organisms that move into the population from another population. So, this is a big factor in especially animals. You don't see as much of this going on with plants, although it can happen where seeds disperse into a new area. This is going to typically happen among your larger mammals, birds especially, organisms that can cover lots of distances. This is a big factor. Deaths, obviously, mortality, the number of deaths that occur in the population at any given time. This will vary greatly depending on species. Some organisms have very long life expectancies. Again, like elephants are 50 to 60 years in the wild, whereas mice maybe less than a year. Butterflies, right, the whole life cycle might be a few months, with actual butterflies live only maybe 2 weeks. So, and environmental factors, of course, play a big role. So that's going to vary quite a bit. Obviously, human influences can really increase deaths in the form of hunting, or could be mass commercial harvesting. How about emigration? Organisms moving out of the area. Again, that's going to be a bigger factor in our birds and larger mammals. So these are all the factors, the basic factors that affect growth rates. Now, you can see we mentioned several times we're discussing death and death rates and immigration, emigration, etc. That lifespan is a big factor. It's going to vary greatly by species. Species are genetically wired to live a certain length of time. The maximum lifespan is the longest period of life reached by a given type of organism. So bristlecone pines, 4,600 years. At least in recorded history, documented, we have humans living up to about 120 or slightly over 120 years, although you hear all kinds of different claims out there. And then microbes may only live a few hours. So this is going to have a huge impact on how your population grows. If you have slow growth, you live a long time, your generation time slows down, population growth can then also slow down. And then, microbes may live a few hours, but in that period of time, they'll reproduce. And so with the short lifespan, you have rapid reproduction. Things grow very quickly. Differences in relative longevity among species are shown in survivorship curves. So we will look at a few different curves that describe organisms that have short lives where they reproduce quickly and those that have long lives and it takes a long time for them to reach sexual maturity. For example, elephants don't even reach sexual maturity until they're between 18 and 20 years, whereas a cat can reach sexual maturity in only 6 months. And a mouse can reach sexual maturity in a few weeks, as well as rabbits. So this is going to really vary tremendously, and that's all determined by the genetics of the organism. So let's now move on to some of the survivorship curves that we have out there. So we've got several survivorship curves out there, and this is going to tie into our topic that we had talked about, R and K selected species. So let's look at the three general patterns of what we call survivorship curves. A survivorship curve is where we graph the life expectancies and basically the population over its age period, over its time. So for humans, you would see a group of, say, humans we're tracking, and let's say we're looking at a group of people all born in 1940. Okay, so looking at all the folks born in 1940, and we'll see, of course, there's a very high number because they were all just born. It might be a slight drop because of infant mortality, but then it kind of levels out for a little while. Not many children are dying here, right? And then it starts to decline a little bit more, and then they start hitting kind of that later season of life, and it starts to drop off really sharply, and say that's about 90 years old right here. So at 90, there aren't too many folks still alive there, and then 80, and then say on down, this is say around 50, etc. So this is a survivorship curve for human beings. Notice, there's not a huge drop-off for a long time, right? Most of us live to be, say, 50 years old. But then it starts to really decline sharply as age, diseases, and things like that start to increase. And so, a fair number of people live to be 80, but then very few live to be 100, let's say. So basically, you have your full physiological lifespan if the organism survives childhood, if you get past that. And that's what we see in elephants and bears, and also humans. So we typically live our full lifespan. If we can just get through childhood, usually we do pretty well. And then death rates greatly increase as we get older. Elephants start to die off really quickly because their teeth actually wear down. Their tooth enamel wears out. They can't even eat once they hit around 50 years old. And 60, they start losing a lot of weight, and they actually die of starvation, not because there's not enough food, just can't chew it. Got to break it down. And so sometimes they'll sedate an elephant and sharpen its teeth up, and then an elephant can live a lot longer. Bears are like that. So humans, we're all kind of in that same category. And this is what we call a type 1 survivorship curve. Next, we have probability of death rate is unrelated to age. So in this type of death rate, or survivorship curve we might call, we see the graph looks kind of steady. So it just goes down at a steady rate. So you're just as likely to die when you're a few weeks old as you are a few years old as you are many years old. You're always, your likelihood of death is just basically consistent all across your age. We find this is the case among gulls, mice, rabbits, a lot of invertebrates like crayfish, although they're probably, actually crayfish would be more like the next one. But anyway, mice, small birds, that's a great example. Again, gulls. So any of your songbirds, they're just as likely to die when they're young as when they're old. And so it's consistent. So you get kind of a consistent curve, and this is called a type 2 survivorship curve. And then we have type 3, where mortality peaks very early in life, and that's true for trees and fish. So what it looks like there is you see a huge amount of death early in life, and then it starts to kind of level out. And again, this is true for organisms that don't care for their young. Like trees don't care for their young too much. Fish don't either. and crayfish, anything that like lays thousands and thousands of eggs, like a clam. One female clam can lay 1 million eggs, guys, and a female tuna fish can lay 30 million eggs. How many of those eggs actually survive? Not many. So they have the type 3 survivorship curve. So again, this is all genetically wired, these organisms. So here are the different curves. You could see the A, that would be the elephant, survived to old age. Notice the sharp drop-off. B, die randomly all throughout life. Again, you're just as likely to die when you're young as when you're old. True for small mammals and small birds and things like that. And then adult lifespan, long adult lifespan, like with trees. So many trees won't make it. Seeds just don't get a lot of nurturing. But if they can get to be large, they usually live a very long time. Okay? So there are your 3 curves. And, of course, this is all genetically determined. Although there are some environmental factors as well, but mostly this is genetically determined. Okay, so let's look at other factors that regulate population growth. So we looked at births and deaths, immigration and emigration. What are some other issues that might affect population growth? Well, we have intrinsic factors. Operate within or between individual organisms in the same species. So, for example, competition between other individuals of the same species, all eating the same thing, all competing for the same resource, maybe it's territory. That's intrinsic. Extrinsic, imposed from outside the population, that might be a predator. That might be human influences. Biotic factors caused by living organisms. They tend to be density dependent. So what do we mean by biotic caused by living organisms? Well, predation, right? That would be an example of a biotic factor. It's a predator coming in, attacking, and eating individuals in the population. Disease is a biotic factor caused by a bacterial species, let's say, or a fungus. And notice it says density dependent. If you recall, density dependent means that the factor becomes more prevalent or severe as the population density increases. So when would disease be worse? When populations are dense or not dense? Yeah, when populations are dense, right? The denser the population, the easier the disease spreads. If the population is spread out, the disease doesn't spread as easily. Abiotic factors caused by non-living environmental components like drought, floods, right? Rainfall, storms, they tend to be density independent, right? For example, whether the hurricane comes or not, right, has nothing to do with your density. You could have a very dense population, the hurricane will come. Your population may not be dense, the hurricane comes anyway. It doesn't really matter, and the hurricane comes and kills a lot of individuals. So abiotic factors caused by non-living environmental components, again, they tend to be density independent. The weather just doesn't care how dense your population is. All right, the drought's going to come whether the population is dense or not. Okay, so all these factors are going to cause the population to be healthy or not. Let's look at this interesting density-dependent factor of predation. Now, we mentioned predation, again, disease, things that are typically biotic factors are called density-dependent, right? Dependent on the density. And so predation's a big one. So let's check this out. We got the snowshoe hare and the Canada lynx. The lynx just loves those snowshoe hares. And there's always a predator-prey interaction going on, in their populations. And they've been tracking hare and lynx populations because we collect their pelts, and there are nice records that people have been keeping track of in Canada and sometimes in the northern United States. And you can see when the snowshoe hare population spikes, typically the lynx population then follows. At least that's the normal situation. So notice here in the graph here, you see the snowshoe hare population right here, guys. Notice how it really peaks, and then right afterwards the lynx population peaks. And then when the snowshoe hare population crashes, well, just right afterwards the lynx population crashes. Notice how the hare is always slightly ahead of the lynx. Well, in this particular picture, right, in the predation, yeah, the hare is going to be ahead of the lynx because the lynx is chasing after the hare. But even in the population graph here, you can see there's a lag where the peak of the hare always precedes the peak of the lynx, and when you have a trough with a hare, the lynx population troughs down too. That makes sense, right? That's a predator-prey interaction, and you can see it's very density dependent. The lynx population is extremely dependent on the density of the hare population, and the hare population is influenced by the density of the lynx population. So they definitely have an interaction. It's a great example of a density-dependent factor. Really kind of neat. I love looking at graphs like that. So we reduce population size by decreasing natality or increasing mortality. And of course, this whole graph was about increasing mortality. We also could reduce the size by decreasing natality, where we have fewer offspring. That typically happens again with the lynx population, because as the snowshoe hare population decreases, there's less food, and the lynx then doesn't have as much energy to produce babies, and so natality drops as the snowshoe hare population drops as well. All right, so we have interspecific interactions between species. Again, great example is predation. Intraspecific interactions will be between members of the same species. Again, predator-prey oscillations is what we call that. Okay, let's now move on to the next slide, and let's erase our marks here, and let's look more at density-dependent factors. Again, we're looking at intraspecific and interspecific. We focused really so far on inter. We looked at an example. Let's look at intra. Competition for resources by individuals within a population. So, among members of the same population, they're going to be competing for mates, for territory, for food. And as the population density approaches carrying capacity, one or more resources becomes limiting. Again, that could be food, it could be water, it could be nesting sites. Control of access to resources by territoriality. One of the ways to deal with intraspecific interactions is to have a boundary, where owners of the territory defend it and its resources against rivals. You'll see this among a lot of songbirds. You'll see this in the next few weeks with male cardinals. They'll work very hard to maintain their territory against other males. Typically, when you see cardinals this time of year, which is getting in their mating time, you'll hardly ever see two brightly colored red male cardinals near each other. They're competing intensely for territory, and once they have a territory established, then they have mating rights, I should say. We see this also with elk. If some of you went to the Ecology Valley field trip we do every year, you saw this incredibly intense territorial battles. Stress-related diseases occur in some species when conditions become overcrowded, like blue or black tongue disease wiped out a large deer population here in North Carolina just a few years ago, and still continues to be an issue. It's a density-dependent factor, and it's typically among members of the same species. So we even see studies where mice even deal with stress-related diseases when they become densely populated. All right, so guys, let's look at conservation biology, and let's look at just kind of take a peek at this topic. So in conservation biology, we are concerned about trying to maintain a minimal population size of a rare species for long-term viability. We could even be trying to cause a population to not just maintain the minimal levels, but to really increase to appreciable levels. We tend to, in conservation biology, as we try to preserve biodiversity, we tend to look at the world through this lens of what we call island biogeography. So if your goal is to maintain a minimum population size of a rare species, if your goal is to maintain biodiversity, you tend to see the world as small islands. And what I mean by small islands is small islands of habitat. They could be actual islands, or they could be patches of woods, for example. So if you look at an aerial photo of Burke County, you'll see patches of habitat and then interrupted with patches of human development. And so small islands, we've noticed, far from the mainland, so actual islands in the ocean that are far from the mainland have fewer terrestrial species than large, closer islands. And so MacArthur and Wilson expanded the idea proposed that species diversity is a balance between colonization and extinction rates. So, for example, if this is the mainland, so there's the mainland, I'll put an M for mainland, and we have an island that's really close by, you could imagine that every so often you'll have colonization coming in here because it's not that far to go. And this little island that's not far from the mainland will have a reasonable amount of biodiversity. Contrast that with an island way out here. What are the odds that an animal or bird or whatever it may be, well, of course, a bird is an animal, say a mammal or a bird, could go from the mainland to this island way out here? Probably not going to happen too often. So we would expect this island that's further from the mainland to have fewer numbers of species, less biodiversity because you're further from the mainland. Another issue is the size of the island. If you have a big island that's very close to the mainland, we expect biodiversity to be especially high here, versus a very small island far from the mainland would have the least biodiversity. And that's the idea of MacArthur and Wilson. So diversity is a balance between colonization, like from the mainland, and extinction rates that occur on the actual islands. And the smaller the island, typically the higher the extinction rate because there's fewer resources. So let's look and explore this topic a little bit more. In a large population, genetic diversity tends to be preserved, tends to be high. So on the mainland, we would have, a large amount of genetic diversity. On that large piece of forest habitat, let's say, it's kind of like the mainland, we would have lots of biodiversity. So does anybody know what the habitat mainland would be here in western North Carolina? Yeah, it'd be the Great Smoky Mountains National Park, Pisgah National Forest, right, our large areas of forest that cover the mountains. That's going to be kind of like your mainland. And then the islands are our patches of habitat around Morganton, around Asheville, etc. Now, a loss or gain of few individuals has little effect on the total gene pool when you're dealing with a large population. So if we're talking about the large population in Pisgah National Forest, where we have high genetic diversity, hey, you can lose a few individuals here and there. It's not that big a deal, right, guys? But in a small population, let's say a little patch of habitat, maybe a little 20-acre forest surrounded by a golf course in Hickory, that small population can be greatly affected by just small events, and that can have a huge impact on the gene pool. This is evidenced by something called genetic drift, which is a change in gene frequency due to a random event. So, for example, let's say we have this little patch of forest, and we have, dark individuals. Let's just say they're rabbits. So here's the dark rabbits here. And then we have a bunch of lighter colored rabbits kind of mixed in here. Okay? And then let's just say randomly this piece of the forest burns down, and the dark rabbits just happen to be over there. Now you just so happens that all you have are light colored rabbits. And so the whole population has changed now. You've lost the dark colored variety. So all it took was a little fire, small event over there, which basically wiped out the black rabbits here, the dark rabbits. And so you see a change in gene frequency. You have lost the gene that codes for dark color. And now you only have the genes that code for light color. Now the population is more vulnerable because it has lost genetic diversity. But if you had a huge forest and you have a forest fire, of course not the whole forest is going to burn down, just a little piece, and you're still going to have dark and light individuals. That's the idea here, guys. Okay? So genetic drift, let's look at that topic a little bit, and that's actually what we've just started talking about already. Genetic drift, one example of this is where we have the founder effect. It occurs when a few individuals start a new population. So, for example, let's say we had a population of rabbits. Let's go back to our rabbit example. And they're found in this, say, large piece of habitat, and there's a smaller piece of habitat over here. And let's say we've got dark individuals right here, and we've got light individuals. And it just so happens that some dark individuals expand and move to this habitat over here, this small piece of habitat. Now this little piece of habitat, all it's going to have are dark individuals. Just by chance, it just so happens that dark individuals expanded, and so You still have dark individuals over here in the main piece, but on this little patch, everything is dark individuals. That's the founder effect. Just a few individuals starting a population, that has a big change in your genes. Demographic bottleneck occurs when just a few members survive catastrophic events such as natural disaster. We see this in the cheetah population. Somewhere in the past, we don't know when, cheetahs went through a bottleneck. How do we know that? They have very little biodiversity, okay? Very, very little biodiversity here in the cheetah population, because in the past, cheetahs were, here, let's see, guys. Let's see. I don't know if I'm getting a notice that we've lost our connection. I don't know if things are recording or what. Well, look, I'll just continue on, and we'll see how many slides we got left, and we'll go from there. So bottleneck. So the cheetahs at one time, we know went through that bottleneck. And how do we know that? Because the cheetah population, first of all, is small. And when we've done genetic studies on cheetahs, we found that they are all really genetically similar. In fact, we think all cheetahs that live today all descended from one male. Can you believe that, guys? One male is the great-great-great-great-great-great-grandfather of all cheetahs today. And so because of that, cheetahs have almost no biodiversity. This makes them extremely vulnerable. A lot of people think the cheetah population is on the brink of extinction even because of the lack of genetic diversity. We don't know what caused the cheetah population to get so small, but they got down to just one male probably. Wow, that's the bottleneck. Founder effects and demographic bottlenecks reduce genetic diversity. So you can imagine, back up toward the top here, this population of dark-colored rabbits in this little habitat where those dark individuals migrated out. Again, some stayed behind, but those that did migrate out are very vulnerable. Genetically, this population here is not diverse. ND for not diverse. Same thing with the cheetah population. Each may result in inbreeding due to small population size. This has occurred extensively in the cheetah population. There's been lots of incest in the cheetah population, lots of inbreeding. And, of course, there'll be a lot of inbreeding up here in this little patch of habitat here at the top of the screen because it's so small. And the only way to avoid inbreeding is for some of the individuals to return back to the main piece of habitat. So genetic drift typically does not help populations. Inbreeding can then lead to expression of recessive genes that have deleterious effects on the population. So most genetic diseases, guys, are recessive, which means they're hidden, if you will, by normal genes. Well, if you mate with a member of the family, like brother and sister or cousins, then you'll start to see those recessive diseases begin to appear because you have the recessive disease. Your cousin has the recessive disease because you're related, and they begin to mate, and then you start seeing those diseases appear. So all kinds of bad things happen here. Here is a great example of genetic drift. You can see we've got a population represented by beads, and you can see there's the parent population, lots of yellow and blue beads. Just so happens that mostly blue individuals came out. The bottleneck drastically reduces the population, and the only ones that survive are mainly blue, almost no yellow. And so in subsequent generations, you almost only have the blue variety, only a few yellows survive. See how this population, represented by this bottle, has much lower genetic diversity compared to the original population, which had high diversity. Again, that's all genetic drift. Okay? All right, let's move on now to population viability analysis. Minimum viable population is the minimum population required for long-term survival of a species. You have to have a minimal viable population to maintain genetic diversity and to provide constancy and resiliency in the population. Good example, grizzly bears. We've had about 100,000 in 1800 now to only 1,200 today. The species range is just 1% of what it once was. Amazing. They used to cover West Texas and the western parts of the Midwest, like western Kansas. Most of Nebraska was covered. Grizzly bears actually roamed way out in the Great Plains. They are found in almost every western state. Now they're only in limited areas of Wyoming and Montana and parts of Idaho, maybe extreme northern Washington state. Very, very limited. And of course, a large part of Canada and Alaska. It is very fragmented into 6 separate populations. 6 separate populations kind of scattered across North America and the American West and the Rocky Mountain region. We need to know how small the bear population can be and still be viable in order to save the grizzly. We don't know what that number is. Do we need, if there's only 1,200 individuals now, do we need 400 to maintain enough genetic diversity? What's that number? We don't know. We don't know what the minimum viable population of grizzly bears is. We know they're stable, but they're very fragmented. Again, imagine that 1,200 is split down into 6 separate populations down here in the United States. Again, mainly because of habitat destruction and overhunting. Also loss of prey species because we overhunted their prey as well. Metapopulations are connected populations. So we have, let's say we had our 6 grizzly bear populations. So here's our 6 grizzly bear populations spread out across the western U.S. Let's say they were allowed to be connected. So through habitat corridors, like, I don't know, a corridor of riverine habitat or riparian habitat, they're able to go back and forth. Now we have some overpasses for them, some way that they can actually go back and forth from the different patches. That would be called a metapopulation. Unfortunately, that is not true of the grizzly bear population. Grizzly bears are not a metapopulation because they're not connected. But in a normal metapopulation, a collection of populations have regular or intermittent gene flow between geographically separate units. We call that gene flow, when individuals can go from one population to another and intermix their DNA and mate. The source habitat is the place where you have most of your births. Birth rates are higher than death rates. Surplus individuals can then migrate to new locations. So you might have a source habitat here. Maybe this is the main habitat here, this larger piece. And then you have sort of lots of births, and you have an overflow, and individuals can then migrate out of the main patch. Our source habitat around here would be the Pisgah National Forest, right? The mountains, lots of forest habitat there. And then a sink habitat would be like a small patch of woods a couple miles away from Pisgah, where birth rates are less than death rates, and the species would disappear if not replenished from a source. For example, South Mountain State Park. You guys know about the South Mountains. That's a sink habitat for bears. So bears occasionally do come into South Mountains, but they don't typically stay there permanently. So the source habitat is the Pisgah. They roam across I-40, believe it or not, every once in a while, especially at nighttime. And then they wander into the South Mountains, and they can live there for a time, but the birth rates are less than death rates. And basically, the South Mountain State Park area doesn't seem to be able to support a sustainable bear population. So it's a sink habitat. So we have to have source habitats close to sink habitats. Otherwise, the sinks will no longer exist. So let's look at a good example. So here's your source habitat in blue, and you can see birth rates are very high. Birth rates are higher than death rates, so the population is typically growing here in the source, and then individuals are emigrating. And as they emigrate, that might cause the population to stabilize. But you can see the source population is supplying individuals into these sink habitats. So this is definitely a sink down here. And why is it a sink? Well, not enough habitat, maybe lots of hunting going on in here. Just for some reason, this area, birth rates are less than death rates. So more death happening here in the sink. Again, probably a hunted area. Maybe this is some type of a preserve in here. Okay? So Great Smoky Mountains National Park is a great example of a source habitat for black bear. Okay, guys, I think that is about it. It looks like we may have had some technical glitches, so I apologize for that. I'm not sure what it's going to sound like. I haven't heard it yet. But let's go ahead and wrap up with this interesting case of extremely high populations of a crab living on Christmas Island. All right, guys, this island was literally overrun with the Christmas crab, amazingly high population. And just an example of how populations can get huge. I wish I had better footage of just how great the numbers can get. I don't know if they're going to get into it as much here, but it is pretty fascinating. Anyway, guys, look, we better wrap it up. This is Online Lecture 4D. Again, it's wrapped up all of the rest of Chapter 6. Hope you guys have enjoyed this topic of population ecology. Bring your questions to class or email me at mspath@wpcc.edu.